So, what should I say about Aaron? You're going to laugh when I say this. Aaron's my favourite presenter. It's true. But actually, Aaron is everyone's favourite presenter. And I don't know whether you actually really know this, but at the end in our conference, we actually have a lot of presentations to make. Some of you will win presentations. Some of you, like our lecturers, will win presentations. We have a trophy for the guest presenter that you choose as a best presenter every year. And Aaron won it last year.
Wow.
Oh, I should say, congratulations. And shake your hand. But anyway, yes, so this is a hotly contested thing. And you know, because Mark was last week, Mark and Aaron invariably are one of the winners of this trophy. I think maybe it is
Mark's won it twice. That's my third. Just say.
And I mean, I know some of you have had Aaron as a lecturer in physiology, so you already know he's a class speaker. He's also a class cancer biologist, and you're about to find that out today. Thanks, Aaron.
Thanks, Ellie. Hi guys, good to be here and to talk to you about just one small piece of my research interest. So as you can see from the slide, most of my career I've been really interested in melanoma biology. It's an important cancer for Australians. And given that this week your focus in your content has been on DNA repair, which is really crucial when we start thinking about cancer, it's a hallmark aspect of cancer development because, of course, our DNA repair capacity is what keeps us alive and prevents us developing cancer at a very, very early stage. So I have an interest in DNA repair as well, so I thought I'd deliver this talk to you guys today. I always like to throw this slide up at the start. I did my postdoc in a place called Cambridge, a very lovely place. That lovely pub, I spent a lot of time in that pub, it's called the Eagle, and it's sort of a little bit famous amongst bioscientists because this is where Jim Watson stood up on a table in 1952 and announced to all and sundry that he had found, or he and his team, particularly with the help of Rosalind Franklin's X-ray crystallography, had found the key to life. Okay, very much a showman. And I like to put this up because as a scientist, a lot of my research is basic research. And we hear a lot about translation these days, and I'm pretty sure no one turned around in 1952 and said, how are you going to translate that, Jim? Okay? So understanding our biology is crucial, even if perhaps the translational aspect isn't always going to be readily apparent. Okay? Knowledge is power. So melanoma is a big issue in Australia. Now, sadly, Australia and New Zealand, neck and neck, we can claim a very unauspicious title of claiming melanoma as our cancer. Okay, we lead the world in the incidence. And it's not surprising, as this slide attests. We live close to the equator. Our genetics is probably not ideal for that. A lot of fair-skinned individuals and a lot of UV exposure. So when it comes to who is likely to get melanoma in their lifetime, it comes down to three key things. One will be your pigmentation. Okay, very fair-skinned individuals with red hair are much more susceptible to UV damage. Less protected, but that's certainly not to say that darker skinned individuals are totally sort of off scot-free, but less susceptible. The number of moles you have, your nevi count, is a great predictor of your susceptibility to developing melanoma. And that's not necessarily because it will develop in a mole, but there's part of that genetic program that gives you a propensity to go on and develop melanoma. And then, of course, it's your UV exposure. And tanning beds are not, as it often used to be marketed anyway, a safe way to tan. There is no safe way to tan. So your melanocyte, which is the precursor cell for melanoma, exists to provide that pigmentation protection for you. And throughout its life, it's encountering all of this UV radiation, UVA and UVB, and a lot of damage ensues. A lot of reactive oxygen species are initiated, and then that can lead to the acquisition of mutations, and then the loss of tumour suppressors and the loss of senescence mechanisms can lead to malignant transformation. And now you've got a malignant cancer on your hands. Now, I can't really speak about this, I don't really have time, but what we know about melanocyte development really speaks to why this becomes such an aggressive metastatic cancer once it reaches this point. So a very aggressive cancer. So this is DNA Repair Week for you. You're thinking about this, and to protect ourselves, we've developed a whole suite of very sort of finely choreographed DNA repair mechanisms to address the different type of damage that our genomes are going to encounter throughout their lifetimes. So different pathways that use different proteins and mechanisms in this very choreographed repair kind of dance that they do to address these issues and protect the life of that cell. One of the major interests, before I move on to the DNA repair part, is understanding this process and particularly what drives metastasis. And I've always been interested in transcription factors because they regulate all of these programs that determine many of these behaviours. So I'm very interested in any of my 508 students in the room will be familiar with this because I built a prac class last semester around it. And I have an interest in how this might affect things like tumour metabolism, help drive progression. But let's start what was the start of my journey towards understanding DNA repair and its relevance to cancer, and particularly melanoma. And it started with this G protein coupled receptor called MC1R. I was working in a lab who was working on the genetics behind pigmentation, skin, hair, and eye colour. And a lot of the variants in European-derived populations in the MC1R gene that are responsible for what we call the RHC phenotype, the red hair colour phenotype. Fair skin, very UV sensitive, and much more susceptible to the development of melanoma. Now, a lot of work had been done over the years in other laboratories that had come to the realisation that MC1R wasn't just regulating pigmentation in melanocytes, and there's different types, there's dark pigments and then there's yellow-red pigments, hence the hair colour, but also it is linked to DNA repair. So this is a really important pathway regulating the ability and augmenting the capacity of melanocytes to repair DNA. And if you take these, what we call a wild-type receptor producing the darker pigments, we have less CPDs and 6,4 photoproducts. These are different types of UV-induced DNA lesions. A lot of less oxidative damage, which is what the 8-oxo-DG is, less reactive oxygen species, less apoptotic burden. Whereas in the RHC variant melanocytes, we see an increase in all of these things. But at that juncture, this is kind of mid noughties, no one really knew what the mechanism was. How was this perhaps being achieved? Now, this is where I came into the equation. I was interested in a particular subset of nuclear hormone receptors, part of the nuclear hormone receptor family. Now, you've heard of nuclear hormone receptors: estrogen receptor, androgen receptor, thyroid hormone, vitamin D, retinoids. They're all nuclear receptors, and they're all transcription factors. So they bind to DNA and they regulate the expression of genes. That's their business model. Now, I was interested in these guys here, NR4A1, 2 and 3, and I'll use those names, but they can be called NR77, NOR1 and NER1. And these are called orphan receptors because we don't really know what they bind to. So unlike the endocrine receptors over here that bind to a very distinct ligand or a hormone and regulate transcription in response, we don't really know what we want to be regulating these guys, if there is something regulating them. So just a few features about how these guys work, which would be important for some of the things that I'll bring up in this talk. To be a nuclear receptor, they all have a very sort of distinct standard structure, this very variable N-terminal domain, a very highly conserved DNA binding domain, and then this ligand binding domain, which is the part that binds to the ligand in response. Now, importantly, on the end of this is a helix that moves, and that really dictates the activity of the receptor. So you guys have been making GFP fusion proteins, and you've been putting your fusions on the C-terminal end. I've made a whole lot, and they're in this talk, GFP fusion proteins with the nuclear receptors, but I have to put mine down on the N-terminal domain, because if I put a great big GFP on the C-terminal end, that helix 12 wouldn't be able to fit appropriately. So that dictated my choice there of where to put my GFP. Now, when these things bind to DNA and they're not bound to a hormone, they will repress gene expression, the classic endocrine receptors anyway. When they encounter the hormone, they boot off things called co-repressors and take up co-activators, and now they activate expression of transcription. And it's all importantly about that movement of that helix 12. That's important. And it's illustrated here in blue here in the structure, and you can see how with a ligand attached, it moves into a different position. There's also a few very interesting conserved phosphorylation sites throughout the receptor, and I'm highlighting this one because I'm going to bring that up a little bit later as well. So this led me to my first question, okay, and was there a relationship between MC1R signalling and the NR4A genes that I was interested in? So I decided to see that if I treated some melanoma or melanocyte cells with MSH, which is the ligand, the hormone that activates MC1R, to see if the NR4A gene expression was affected by that. And indeed it was. We saw this very robust and transient induction of all three family members in response to that treatment, including with MSH. This is real-time PCR. That's B16 melanoma cells. We also had a large library of primary human melanocytes that we genotyped for the wild type or the variant MC1R. And what fell out of this was that, just like we'd seen in our B16 melanoma cells that have a wild type MC1R receptor, we got induction when we treated our cells with MSH, melanocyte stimulating hormone here, okay, in 3 different strains from 3 different individuals. But when we did the same experiment in strains that were homozygous for these RHC, red hair colour variants of MC1R, we did not see the induction of the NR4As. Okay, so this sort of gave us a little bit of a clue. So the answer was yes, they're rapidly and transiently induced in response to that signal. So why might this receptor induce these receptors, these transcription factors? So that led to sort of the next question. Given that we knew that MC1R was also important not just in pigmentation but also regulating the DNA damage response or augmenting DNA repair, if we take the NR4As away from these cells, how is that going to impact on the ability of that MC1R pathway to facilitate this DNA repair? So to do that, we took some of our primary melanocytes. We used siRNA to knock down NR4A1 and 2 because in the literature they're often redundant to each other, so we hit them both at the same time. We UV irradiated to induce a whole lot of DNA damage in these melanocytes. We treated with the melanocyte stimulating hormone or vehicle, and then a number of hours later, up to 24 hours, we looked at how much DNA damage was remaining in the nuclei of those cells using immunofluorescence. So we don't just have antibodies to detect proteins, we can also detect a particular type of damage in the DNA. And in this case, it was the CPDs, the cyclobutane pyrimidine dimers. It's a type of lesion that the UV radiation induces in the DNA. So what we saw in our negative treated cells was our vehicle cells still had quite a bit of damage left over after 24 hours, but our MSH treatment, you can see the intensity is lower. That is because there is less damage. Now we can also do something called a southwestern blotting to quantify that, and we can quantify these images. But this sort of showed what had already been shown in the literature, that MC1R activation aids the clearance of the DNA damage. But when we knocked out the 2 NR4As, 1 and 2, we lost that ability. We attenuated the ability of MC1R to augment the clearance of that DNA damage. Okay, so we lost that. So that's interesting. That provided a link. So here's our answer. That yes, it does appear that it's going to be important perhaps in this DNA repair process. Now, at the same time, we were sort of interested in this other question of how might this UV damage affect the localization of our NR4A protein in these cells? And again, this is where I was using my GFP fusion protein to see where the proteins end up. Now, this is NR4A1, and you can see a nice green nuclei, and it localizes very nicely with the DAPI stain, showing that it is the nuclei. That's in the no UV condition, but when we hit them with UV, they form these very distinct nuclear foci. NR4A2 did the same, and NR4A3 did the same. They all formed these foci. Now we tested, there's 48 nuclear hormone receptors. We tested a whole lot of others like PPAR gamma, estrogen receptor, androgen receptor. No other nuclear receptor did this. So this seems to be something that only the NR4A family would do. So that's our answer. They all seem to form these foci. Okay. So the next question is, then what are these foci? Okay, it's very distinct, it's very reproducible. Given that there's a knowledge here of the association between MC1R and DNA repair, the logical place to start looking were DNA repair proteins to see if perhaps they were localizing with DNA repair proteins. And we found a lot of proteins, a lot of immunofluorescence data showing what NR4A2 in this case, which is the first one we started following up, was not localizing to. Okay, hours, weeks, months of work just to show what it wasn't doing. But of course, nucleotide excision repair is the main pathway that is going to be deployed by your cells to repair that particular type of lesion that the UV damage causes, the CPDs and the 6,4 photoproducts. Okay, and you can see there's lots of different proteins that come in this choreographed dance to basically allow that repair to occur. So here are our cells, and here are two of those proteins, XPC and DDB2. You can see them here, okay, in the nuclei of cells before irradiation. After irradiation, you can see now that we've got our foci with our GFP and our 4A2 fusion proteins, and we get very nice co-localization, which is why the overlap comes up yellow here, with our DNA repair proteins. Okay, so here we have our answer that it appears that after UV, NR4A2 protein is co-localizing with nucleotide excision repair proteins in these foci. Okay. That's interesting. Now, knowing a little bit about the biology and how these receptors work, I was interested in what domains are responsible for that, and I haven't got the data here. I took it out in the interest of time, but we know that the N-terminal domain is crucial for the foci formation. If I lock that N-terminal domain off, it will not form foci. So the DNA binding domain and the ligand binding domain on their own will not form foci. And in fact, I said other nuclear receptors don't form these foci, but if I swap the NR4A N-terminal domain for the N-terminal domain on one of those other receptors that don't form foci, they now form foci. So there's something going on in that N-terminal domain that is crucial for that foci formation. But of course, there's a lot of functional activity in this ligand binding domain, and I highlighted this helix 12 here. Just for those watching at home, I'll use the mouse here just so you can see what I'm pointing to, helix 12. It's really important for the ligand binding domain function. So I took the wild type receptor, they're fully intact, and here are our foci and co-localization between U2 and XPC. When I remove helix 12, our GFP fusion proteins will still form foci, but they will never co-localize with the DNA repair proteins. Make a great Christmas card. Lots of red and green, but not a lot of yellow. Now, biologically, I know that what is happening when we lock off helix 12, we're making a dominant negative receptor as a transcription factor, because it is going to bind to co-repressor, and it is going to close down chromatin because that pulls in histone deacetylases, and it deacetylates the chromatin. If you know how transcription works, deacetylating the chromatin will shut it down. Now, to facilitate transcription or to facilitate DNA repair, you need to open that chromatin up. You've got a lot of big proteins that need access to that damaged DNA. And so here's what I think these guys are doing. So we treated these same experiments with trichostatin A, which is a chemical inhibitor of histone deacetylase activity. Lo and behold, in most cases, we got a pretty good rescue of the phenotype, and we now got co-localization. So this started to help us get a handle on a potential mechanism here, that these receptors are forming foci at sites of DNA damage, allowing recruitment of DNA repair proteins to that site. If we tinker with that and we shut that down, then those repair proteins aren't going to gain access to those sites. So our delta helix 12 mutant definitely disrupted the ability of these foci to form a co-localization, but not foci itself. We also looked at double-stranded DNA breaks as well. Okay, so we treated with UVB and we looked at markers for double-stranded breaks. There's a number of different ones. I'm just showing you 53BP1, and you can see over a time course the clearance. So the question here was, if we overexpress wild type or different mutant forms in our melanoma cells and then hit with different types of DNA damage, will we affect the ability of these DNA lesions to be cleared, and in this case, double-stranded breaks. So we're doing this by immunofluorescence. So you can see the foci in our empty vector control. You can see when we overexpress the wild-type NR4A2 receptor, we get better clearance. And we can quantify that based on either the number of foci per nuclei on average. You can see you get impaired clearance here with the NR4A2 overexpression, or the percentage of cells in the field that are still foci positive. Now, when we looked at a couple of the other mutants, here's the phospho mutant that I was very interested in, and we can see that that impairs the foci formation, actually less foci formed, but it took a lot longer to start clearing them away. So we disrupted the kinetics of that repair. Likewise, that delta helix 12 mutant that I just showed you, we seem to have statistically significantly less foci per nuclei, but those foci that do occur are not being repaired as effectively as they are in the empty control cells or the wild type cells. So yes, it does seem to be affecting double-stranded break repair as well, not just nucleotide excision repair. Now, I'll just jump through this very quickly, but we also saw this with gamma irradiation-induced breaks as well. So for the last little bit, all of that work's been published and we're very proud of it, but I think there's a really interesting story with NR4A3 here, and we've not published that, and there's still lots of pieces of the puzzle. We've got lots of little interesting sort of bits and bobs in our drawer, but still we haven't formed the whole story. So what about NR4A3? Well, it is also being induced by MC1R signaling, as the other members are. It also forms these foci after UV irradiation. But we also know that these NR4As are not in the same foci. Okay, so when we have our GFP or YFP flagged NR4A3 and a flag tagged NR4A2 and we do immunofluorescence, we get this nice red and green pattern. They're not going to the same foci. So we set about doing the same thing and asking the same questions as we did for NR4A2. And that is, what does it co-localise with? And we started sort of looking through some of the logical candidates, representatives from each of these different DNA repair pathways. And here are some of the ones that sort of popped up with this nice co-localisation.
Ooh, I think if we leave the questions to the end, we'll do our number thing again. Right? Sorry.
So this is UV irradiation, okay, and you're seeing this nice co-localisation with just 3 proteins I'm showing you here, OG1, 8-1 and SC35. Now, what's interesting about these proteins is they participate in a particular pathway called base excision repair. Now, what's interesting about base excision repair is that it's the pathway that's going to deal with reactive oxygen species generated damage, a particular type of lesion called 8-oxo-DG. So with that in mind, we did the same experiment now in the context of high oxidative stress. So we treated our cells with hydrogen peroxide and we saw foci formation, and again we saw co-localisation with these lesions as well. So remember, in UV damage there will be a lot of reactive oxygen species generated as well. So that type of damage will exist. And probably the most important melanoma mutation, BRAF, B600 to glutamic acid, is not a UV signature mutation. We think it's probably an oxidative lesion. So oxidative repair is very relevant to melanoma. So there's our answer to our question. NR4A3 co-localises with base excision repair. It did not co-localise with nucleotide excision repair proteins. That seems to be something NR4A2 did. So this is where we're left with a lot of little pieces of the puzzle trying to work out where our story might go with NR4A3. And I think there's a potentially interesting story here. So is NR4A3 linked to cancer susceptibility via DNA repair? Okay, so what's some of the interesting data we have that sort of might speak to that? I have an idea that maybe NR4A3 mechanistically is requiring RNA processing as part of its functional activity. Now, why I thought that is I was sort of flicking through some journals one day and I came across this paper that talked about histidine repeats in proteins. And they'd done a genome-wide analysis and come up with this idea that proteins with large tracts of histidines, and you're talking maybe 4 histidines in a row, are likely to be involved in the formation of nuclear speckles, which is kind of what those foci I was looking at are, and they'll be implicated in RNA processing. Things like splicing or nuclear RNA metabolism and processing, or interacting with RNA. NR4A3 has 14 histidines in a row in the N-terminal domain. The other family members do not have this. It's unique just to NR4A3. So this got me thinking, well, what's going on here? So the first question I thought, how am I going to address this is, if this is important, what if I took those histidines away and I replaced them with alanines? Okay, will that impact foci formation? So you can see here that the histidines in NR4A3 are fairly unique to NR4A3. They're not there in NR4A1 and 2. So I basically swapped out the histidines with alanines. And it totally screwed up the ability to form foci after UV. So the answer is that, in both UV or peroxide treatment, it did alter the ability to form these foci. Now keep in mind, a large alanine tract like that is going to have a fairly substantial impact on the structure of that N-terminal domain. So that you might just be seeing that sort of disruption of N-terminal domain structure. But what about the potential functionality here? So the question I was now asking is, in light of that, is RNA binding going to be an important part of whatever NR4A3 is doing at these foci and its potential role in DNA repair? So to get at that, another immunofluorescence experiment. Okay, I irradiated the cells, but has anyone ever done immunofluorescence? Okay, you permeabilise your cells, then you fix them, and then you start staining with antibodies. What we did though was we permeabilised, then we treated with RNAs to degrade all of the RNA in the cell, and then we fixed them and proceeded with our immunofluorescence with our primary and secondary antibodies. And so what we saw here was in a vehicle treatment, just PBS, no UV plus UV, there's our foci. And our DAPI staining shows us this is what our nucleus is looking like. When we treated them with RNAs though, we did not see foci. So treatment with RNAs seemed to disrupt and degrade these foci as part of that treatment, which was interesting. And we did not see this with NR4A1 and 2. So when we did the same experiment with NR4A1 and 2, we saw the same sort of foci in the RNase treatment as we saw for NR4A3. This was only happening in NR4A3. So I think there's an interesting mechanism story here, and I love mechanisms. So this is something I would love to chase up down the track. And RNA processing in the DNA damage response is really, really hot. It's a really exciting area. Another interesting piece of the puzzle comes from somebody else's lab, a lab in Texas, that made the NR4A1 and 3 knockouts. And what they found was when they knocked out NR4A1 and 3 together, and they could do this in a sort of a heterozygous fashion, it didn't have to be homozygous knockout, these animals developed very rapidly, soon after birth, acute myeloid leukemia. That's interesting. You lose your NR4A3, you develop a blood cancer. Now, the authors did notice that they, and did note in their supplementary data, they didn't really fixate on it, they cited my work, which was nice to see, that there was an increase in gamma H2AX foci, which is a type of break repair marker, in the lymphocytic cells of the knockout mice. So that begs the question to me is, does the cancer in these mice arise because they've lost NR4A3, therefore they've lost an important DNA repair capacity, and now they're more prone to going on and acquiring mutations because of that disruption in repair? Again, this is something I'd really like to follow up. We also know that NR4A3 is linked to extraskeletal chondrosarcoma, a pretty nasty soft tissue sarcoma that always arises due to a chromosomal translocation that creates this Frankenstein protein, which is a fusion protein between the full in-frame NR4A3 open reading frame, so everything comes along, full N-terminal domain, DNA binding domain, the hinge, and the ligand binding domain with the all-important helix 12, and it's fused most often to Ewing's sarcoma, or part of the Ewing sarcoma protein, but also a number of other proteins. But the NR4A3 part is always intact. Here's an interesting little tidbit, and I do not know what this means, but the part of these proteins, Ewing's, RBP, TCF, and TFG, that is fused to the N-terminal domain of NR4A3 is an RNA binding domain. What does that mean? you think it might mean. Okay, we've already got this idea ticking away that somehow interacting with RNA is going to be a really important mechanistic part of whatever NR4A3 is doing as part of DNA repair potentially. And now we've whacked on another RNA binding domain, so a type of domain in a protein that will interact with RNA. I don't know what it means, but it's very intriguing. It's certainly tweaked my interest. So we, as we had done, as I've shown you previously with our GFP fusion proteins for NR4A1, 2 and 3, we created this fusion protein and then put a GFP tag on the N-terminal domain. And lo and behold, this fusion protein, because I thought, well, maybe it stops them going to the foci. Okay, so maybe it disrupts the structure of the N-terminal domain and they don't form foci and therefore it can't participate in DNA repair. Well, in response to UV, hydrogen peroxide, or potassium bromate, which induces a lot of oxidative stress, we form foci with our Frankenstein fusion protein. So they're getting potentially to damage foci, but when they get there, are they behaving the way they should? Getting to the foci is the first part of the job. Facilitating DNA repair is the next part of the job, and is that disrupted? Again, it's another little piece of the puzzle, but again, lots of questions to answer. I also work with a group of geneticists out at TRI that have a number of cohorts of patients that have high-risk melanoma. Now that's predicated on the fact that they're getting melanoma at quite an early stage, and they're also getting multiple individual primaries at different stages. So it fell out of a larger cohort which was looking at nevus genetics. Okay? And that's why you can see on the slide here, CMM means cutaneous melanoma, so we've got a number of patients with a certain number of cutaneous melanoma, and the TNC is the total nevus count. You can see some people have 87, 36, so quite a lot of nevi. So we had 592 patients in the cohort, and also another 161 that had a family history of melanoma, and this was versus 502 control subjects in that cohort that had no melanoma history at all, across varying ages and both genders. And the cohort was then divided into 4 groups for exome sequencing. So our highest risk category are the individuals who were under the age of 40 but had multiple primary melanomas. Okay, not just one, but multiple individual primary melanomas before they even turn 40. That's quite early. That's our highest risk group. The next highest would be the people that are still getting the melanoma under 40, but they've only had one cutaneous melanoma under the age of 40. Following that, we've got people who didn't get a melanoma until after they were 40, but they've had multiple individual cutaneous melanomas over the age of 40, and then the lowest as part of this group are the people that are over 40, but they've only had the one cutaneous melanoma diagnosed over 40. Now, not surprisingly, when the exome sequencing was done, a lot of DNA repair proteins fell out of the analysis. So a lot of variants in very important known DNA repair proteins. And I asked them in the study to include NR4A3 and 1 and 2. They didn't find any mutations or variants in NR4A1 or 2. Okay. Now keep in mind, they're not looking for mutations in the tumour cells, so it's not like these are acquired mutations that are driving the melanoma. These are germline mutations, or not mutation, I should use the word variant. So they're variants in these genes, but potentially deleterious variants that are disrupting the ability of these proteins to function normally. So no NR4A1 or 2 variants were detected in the germline of these individuals. But out of the 600 or so patients in the melanoma cohort, there were no variants in NR4A3 detected in our control subjects, but we essentially found 6 individuals that had what would be predicted to be a disruptive variant, or mutation if you like, in NR4A3. You can see where they fall. Most of them here are in the LBD, but we've got one variant, the P269 to arginine, that is occurring in the N-terminal domain. It's occurring in our highest risk group, the multiple primaries under 40, okay, along with this serine mutation here. And it's also appearing twice in our multiple primaries under 40 group down here. So not a huge number, but out of 600, 6 out of 600, it's still not bad. And that's of course going to be in the context of potential variants in a whole lot of other DNA repair proteins as well. So there could be a cumulative effect going on. So the answer is maybe. But that just leads to more questions. So every answer you get in research is going to lead to another question. It's really just, there's a segue to the next question, and that is, well, do these guys form foci? Do they co-localise with repair proteins appropriately? And will they facilitate or disrupt DNA damage responses and repair? I don't have the answers for that at this stage. So again, this remains a little piece of the puzzle and questions to be answered. And this is, I promise, my last slide to keep on time. This has tweaked my interest because this paper came out a while ago that shows that a particular molecule, dendrogenin A, drives LXR to trigger nuclear autophagy in cancers. Now, LXR, pharmacologists in the room doing pharmacology with me tomorrow, LXR is a nuclear hormone receptor. And what this paper showed was LXR being activated by dendrogenin A induces NR4A3, which induces this autophagy response. And if they knocked out NR4A3 activity, so if they removed NR4A3, dendrogenin A would not induce autophagy via LXR. So NR4A3 is the crucial link here between this activation of lethal autophagy by LXR. Now this opens up a potential therapeutic avenue. We might be able to trigger lethal autophagy in different cancers that have high NR4A3 expression. Now that could be melanoma, that could be the blood cancers, the leukemias that have high NR4A3 expression. So an interesting idea here. So the question you might be asking is, why is that relevant? Well, autophagy is a really important pathway that is going to be protective to cells. So that is why perhaps inducing NR4A3 is part of that cytoprotective melanocortin-1 receptor pathway I started off with might be important, because maybe it's augmenting cellular protection via autophagy. But also we now know that autophagy and DNA repair pathways are very intimately linked. And so autophagy is really important for efficient DNA repair. And DNA damage is talking back to the autophagy mechanisms to engage with it to facilitate that process. So again, I think there's potentially a really interesting little story going on here. But again, lots of little crumbs that are interesting and exciting, but the story is yet to completely emerge. So watch this space. And if anyone's interested in a project like this, I'd be more than happy to talk about honours and masters projects in the lab around this. And throughout I've been saying we did this. Pretty much I was doing the odd experiment, but really I was writing grants and doing all the boring stuff. And it's these wonderful people that have come through my lab over the years, and particularly Kelvin, who did a lot of this DNA repair work as part of his PhD. My former postdoc Yash, who now has his own lab in Philadelphia, contributed a lot. Kasturi, who went on to become his wife. She also started off a lot of the foci work. Mitch was another PhD student who's now got his own lab in Philadelphia as well. He and Kelvin, they basically got the band back together but didn't invite me. But some wonderful people have come through the lab, and it's probably the part of my job I like the most, is mentoring students in the lab, and it's the thing I will miss the most when I finally decide not to do this job anymore. Thank you for your attention. Any questions?
Look at the numbers. What's going? You're going to have to help. You don't have
Do we start with one, or is it
No,
like
they're all randomised.
It's like a bingo system.
So yes, well, not really bingo. Oh. All right. Let's get going. Number one.
So cool. Sorry, looking back on your study of where all your different cohorts, the over 40s, under 40s, and your main one was what had 2 different mutations in it. Your first, MPM. You said there was 6 individuals. So how many was actually in this group?
Around 800, so 592.
So how many with these mutations were actually contained in that top group?
6. So we found one patient with that mutation, one patient with that mutation in that group. So that's only 2 in that particular cohort. We found 6 all up. Out of about 800, I think it's more like 750 melanoma patients versus controls. There were no mutations in the controls, but we found these 6 mutations. So we found 2 individuals with the 269 to arginine change in this part of the cohort. Okay, so 6 all up. So it's not a huge number, but keep in mind that's also in the context of variants that occurring in a whole lot of other pathways. So it's not just necessarily about the loss, the serious loss of one gene. So it's not sort of highly penetrant like, say, the BRCA mutation, although there are BRCA variants that would be predicted to be deleterious to BRCA in this cohort as well.
So did you go on to have a look at what other mutations actually coincided with those?
We have a list and we're working our way through. This is very much a work in progress. This hasn't been published. It's not all my data, like I've logged into this because this was my part of it, the interest, because that's what you start to want to build up is, because keep in mind these are germline variants, these aren't acquired mutations, so it's not like, we found 6 melanoma tumours with these mutations. We found 6 individuals with what the bioinformatics would predict is a deleterious mutation functionally. It is going to disrupt the receptor's function. So there's other proteins that are going to be sort of involved in that.
Aaron's going to try and answer the questions much faster. So many to get through.
I got the environment by son.
Yes. Yes. Number 3. Sorry, Aaron.
Do we not have number 2?
No, I've got 2.
Oh, okay, sorry.
You may have already talked about this and it went over my head, but did you find what was happening in the N-terminal, which was important for the formation of the foci?
Not completely. We know that N-terminal domain is important. In fact, I threw like a little sort of summary slide, and again, I'm going to sort of annoy Sally by throwing that up. But, we've done things like mutate different areas, look at some of the signalling pathways that are using chemical inhibitors of different signalling pathways to show that when we, say, inhibit PARP or inhibit P38, we don't form those foci, even though the N-terminal domain is there, swapping the domain over. I'd really like to know specifically what it might be interacting with, and there's a particular type of experiment I would do that, like an immunoprecipitation experiment, to basically capture what proteins RNR4A 1, 2 and 3 are interacting with to facilitate that, because that's what's going to be happening here. It's going to be a phosphorylation event, most likely, that then facilitates an interaction with another protein that draws it to that site. So, I'd love to go exploring and find out mechanistically what is doing that.
Wonderful.
That's me. So I know that NR4A3 was obviously really important with a lot of these findings. Is there much importance, like would there be more experiments worth doing with the NR4A1 and 2, or would it kind of be more directed to the 3
This
now?
also speaks a little bit to luck in science.
Okay.
It's basically was a complete, I only had so much money, so many students.
Yeah.
So I basically said to Kelvin, 1, 2 and 3, which one interests you the most? Pick one and run with it. And he picked 2. And it seemed to sort of yield data much more easily. NR4A3 was a little bit more difficult to get at. So if he'd chosen NR4A3, he may not have progressed as well through his PhD as he did. So that was pure dumb luck of just saying, 2 looks interesting. He could have picked 1. Now, we have a lot of data on 1, and I still have no idea what NR4A1 is doing.
Okay.
So, lots of interesting data, but still no sense of, is it no association with any foci, no link to any particular type of damage that might susceptible, or the cells would be susceptible to if we tinker with NR4A1. You know, as you've probably seen, what we largely do with these things is we take our genes away, or we put the wild type or mutant versions of them into cells and then see what happens to the cells when we insult them. Genotoxic stresses, UV, oxidative stress, and so forth. So NER77 hasn't been very yielding, so which is NR4A1.
Yeah.
So, yeah.
Oh, thank you.
You said you did the knockout of NA4A1 and 3 in the mice, and they got the acute myeloid leukemia.
I didn't do that. Some guys in Texas did that. Yeah.
Is there any thought of looking into like the NR4A family in the acute myeloid leukemia instead of melanoma?
Yeah, absolutely. What I think this data speaks to is, you know, I've come at this with an interest in melanoma and UV damage and oxidative damage, but I think these guys are going to have a much broader role in facilitating DNA repair. And I think the blood cancer as well speaks to that. The extraskeletal chondrosarcoma speaks to that. I think generally they're going to have an interesting interaction with the DNA repair process, and that will lead to different types of cancers. So we can get a handle on that by doing some bioinformatics, looking at sort of stratifying cancers in the genome atlases that, you know, are high NR4A1, 2 or 3 and low NR4A1. You can see if there's a representation there and a skew towards a particular type of cancer. As you can see, my suggestion here is they're probably associating with different types of repair pathways. So, and that's why different types of cancers are more, will arise more from different types of damage. So therefore, disruption in one particular member might be more associated with a particular type of cancer.
Makes sense, thank you.
Okay, 6.
So for NR4A proteins, you mentioned there's a change in localization. Have you found in literature this change for other cancers as well, or just melanoma?
There's been a couple of other groups that have sort of followed on from us and have shown the same sort of localization. Are you talking for the NR4A proteins themselves? But no, this has been our little sort of niche area that we've sort of plugged away at. And yeah, there was another group that also, it's always nice when other people can either sort of replicate your work or provide data that also supports where you've gone, but also showed an association with strand break repair as well and formation of those foci. But in the DNA repair area, these sort of foci are pretty stock standard. They see these foci around sort of DNA repair regions or areas of active DNA repair.
Right, 7.
Treating melanoma. Are there any like current therapeutics that you could see just for it, or is it still too early?
So as far as the NR4As go, they're orphan receptors, so they're not going to be as sort of amenable to treatment as things like LXR, which is an adoptive receptor. That's the target. But that's why I threw up that last bit, is we could maybe sort of drive NR4A activity, NR4A3 activity specifically via LXR, because we do have drugs that can either agonise or antagonise. One of them. And do that via LXR. So this is why understanding the pathway, the basic science, and building up our understanding of that network might sort of identify ways that we can kind of get at that pathway. That said, there are some small molecules emerging that will interact and modulate the NR4As. And if you speak to Sally, these guys are all stimulated. So, you know, we can kind of come at these sort of proteins a different way. That's always been one of the problems with transcription factors, is they're not easily sort of druggable targets, but they're biologically important. And there are some very funky ways we can get at that, but there's nothing currently emerging to address that.
I forgot what number we're up to. 8.
What medical potential will you further investigate regarding making NR4A3 susceptible to DNA damage?
I'm intrigued by this dendrogenin A. I'd like to know more about how that pathway is working, because that's, again, a druggable target. A lot of basic research is needed to understand this process before we can start thinking therapeutics. But hang on, but that said, NR4A, the nuclear hormone receptors are a fantastic class of drug class. They're number 3 behind, I think, kinases and G protein coupled receptors are number 1. They're fantastic drug targets, so understanding their biology, I think, is important, and will have translational outcomes down the track.
I was wondering, when you were talking about binding to the RNA
whether it was mRNA or DNA. That's
I don't know. I suspect it could be microRNAs, it could be long non-coding RNAs. We know that there's a number of long non-coding regions, RNA molecules in our genome that perform really important functions in transcription, in DNA repair. So I've thought a lot about this over the years, how I might capture those RNA molecules and work out what they are. Not an easy thing to do, but it can be doable, and I've tried to link up with people over the years that have got that technology and do that. So not a cheap experiment, so there's kind of issues of money and time. Yeah, I'd like to know what it's binding to, but I suspect it's going to turn out to be some sort of interesting long non-coding RNA.
All right, number 11.
Did you find out why RNAs disrupt the host site?
So what would be happening there is, we know, with things like these long non-coding RNAs, is that, they're long and they form lots of different loops, and they actually interact with different proteins that have RNA binding domains. And that's actually one of the mechanisms that helps sort of tether them together. So they're induced to bind to this RNA. So this long RNA molecule might be sort of holding 3 different proteins, NR4A3 being one of them, together in that complex. So another part of that long non-coding RNA might be tethered to one of the repair proteins, direct repair proteins like PH2 or also A1 or something like that. So when you treat those cells, before you fix them inside, when you degrade the RNA away from them, then those complexes just sort of fall apart. And then when you fix the cells, all the foci just fall apart, and now you've got this pan-nuclear state.
12? No, 13.
Back in one of your slides, you were saying you found that P269A was found in the end terminal, and that was found in P under the 40, or they were more at risk. But when compared to the P26 in the ligand thing, was there more research on why it was found to have more risk at the end terminal than the ligand terminal?
Just say that last bit again.
Because you have the P269.
So when we, basically all we've done is we've taken patients that have had melanoma and a control group that have never had melanoma. And within that melanoma group, we've just divided up into groups arbitrarily, but by saying if you've got more than one melanoma under the age of 40, you're in the highest risk group. So it's really just sort of saying, amongst our melanoma patients, this is our highest risk to our lowest risk. So we found 2 patients with mutations in NR4A3 in this highest risk part of the melanoma group. But at the end of the day, we still found 6 individuals with mutations in NR4A3 out of about 750 patients in that melanoma group. So they're all in the melanoma group. But it's interesting that this particular mutation we've now found in 3 individuals. It's the same mutation in 3 individuals. Now keep in mind, like in some of the driving oncogenes, things like the BRAF mutations in melanoma, around 50 to 55% of patients have the exact same mutation, and it's the V600 to E change in the BRAF kinase. So, and that's just a signature mutation. So some proteins you end up with lots of mutations and they're all different. Some you end up with a signature mutation. So it just so happens that for some reason we found this particular mutation 3 times in 3 different individuals.
Right, 14.
Hello, I have a general question. I'm quite pale. I've got a lot of freckles and moles. Is there anything I can do aside from sunscreen to protect something on?
Just to be sun smart. Australians are more sun smart than just about any other nation, and I'd say New Zealanders as well. So we're very aware of the limitations of the sun, but that said, we still live in the sun, we go to the beach, we're outdoor loving people. But yeah, it's about protecting yourself from using sunscreens, using sort of cover, not going out in the sun at the sort of peak UV. That's the best you can do. Awareness and checking. We have great screening programs here in Australia that other countries don't have. So just being aware if it's an issue here, and responsible.
So we have no hope of getting through the rest of you. Anyone who can stay will stay outside and answer questions. I know there's an honours information session right after this. You gonna go? Neither am I. I've got to do colonies. Yeah. But anyway, look, Aaron is open to taking up with his master students. So if you think that it's interesting and you can't stay, why don't you send him your email and any questions that he gets, CC me, and then I'll know, right? So.